So today we'll be talking about the questions asked for Viva in a typical Catalase test. Yes, so the first question says, what is the reagent used for the Catalase test? So here the most commonly used reagent is your 3% of hydrogen peroxide, 3% H2O2. But remember, for certain anaerobes, it is around 15% of H2O2. For certain organisms like Neisseria, it may be 30% of H2O2. But most commonly, we use 3% of your H2O2, 3% solution of hydrogen peroxide. The second question reads, what is the reaction catalyzed by this catalase? So catalase, it catalyzes the breakdown of hydrogen peroxide to form water and liberate oxygen, and it is this effervescence of oxygen which tells us whether the organism is positive or negative for catalase. So the breakdown of hydrogen peroxide to form water and oxygen is the reaction catalyzed by the enzyme catalase. What is the role of catalase? This catalase in organism, it prevents the cell which is producing the catalase from the oxidative damage of the reactive oxygen species. So whatever oxidative damage would have been done due to species like hydrogen peroxide which are formed during aerobic respiration, this particular damage is prevented because catalase, it breaks down this hydrogen peroxide to form water and oxygen. Okay, so state some examples of catalase positive organisms. You will find many. You have organisms like Listeria, you have organisms like E. coli, you have organisms like Bacillus, you have organisms like Pseudomonas. There are many, the list goes on. You have Nocardia species which are catalase positive. Organisms which are catalase negative, it includes the species of Streptococci. It includes the species of what you call as anaerobic organism. Generally, the organisms which are anaerobes, which do not respire but which only do fermentation, they are catalase negative. So we have organisms like Streptococci, organisms like Enterococci. Actually, it is used to differentiate between these organisms which are catalase negative. What is the common precaution which we take during performance of this catalase test? Try avoiding using a metal loop. So do not use any metal. So no use of metal loop. You can use a wooden applicator stick because use of metal loop may give you a false positive result. What are the two basic types of catalase test? Yes, it may be done either by the slide method or by the tube method. Some also pour hydrogen peroxide in the slant. So these are some of the common questions which will be asked for the Viva for this catalase test. Stay tuned with us for more description and details and more questions in microbiology.